Good evening, everyone. Welcome to lecture 3 for the series Biomolecules. So in the last lecture, what we studied was, we studied about the proteins and what is the composition of proteins, how proteins are made up of. So we studied that proteins are nothing but they are the polypeptides which are made up of amino acids, and these amino acids are linked through the peptide bonds. And in today's lecture, we will study about carbohydrates and lipids, which are another class of biomolecules, and they also have a they also play important role in the biological functions. So this is how the biomolecules were classified. So biomolecules were broadly classified into carbohydrates, proteins, nucleic acids, and lipids. So carbohydrates are made up of monomers, like for example monosaccharides, which are joined by the glycosidic bonds, and examples include starch and cellulose. Uh, Carbohydrates are made up of elements that mainly composed of carbon, hydrogen, and oxygen. And the functional importance of carbohydrates is that they are the energy source. They are the structural components of various components. They are also the structural components. For example, chitin, we know, cellulose, which is a plant carbohydrate, which is present in plant cell wall. So they also play role in uh, having structural, so they are also the components in many structural entities, and they also act as reserve foods. So starting with carbohydrates, carbohydrates are the most abundant biomolecules on the earth, and each year photosynthesis converts more than 100 billion metric tons of carbon dioxide and water into cellulose and other plant products. Certain carbohydrates, for example, sugar and starch, are a dietary staple in most parts of the world, and the oxidation of carbohydrates is a central energy yielding pathway in most non-photosynthetic cells. So, in most of the non-photosynthetic cells, for example, if we consider humans also, so in case of humans, carbohydrates are the energy source that provides the energy to the cells in our body, and by the process of oxidation of carbohydrates. Insoluble carbohydrate polymers serve as structural and protective elements in the cell walls of bacteria and plants and in the connective tissue of animals. Other carbohydrate polymers lubricate skeletal joints and participate in recognition and adhesion between the cells. More complex carbohydrate polymers covalently attach to the proteins or lipids, so they may be present in the conjugated form, and they act as signals that determine the intracellular location or metabolic fate of these hybrid molecules called glycoconjugates. So basically, the if we are asked that which is the most abundant biomolecules on the earth, so these are the carbohydrates. So from these examples we get to know that from these facts we get to know that carbohydrates play important role in providing the energy to the non-photosynthetic cells. Also, they serve as structural and protective elements in various cells. For example, in case of plants, also they are present in the cell walls. In case of insects, they are a part of exoskeleton, etc. And also they play a role in conjugation with the proteins and the lipids. They also play the role in signaling. So carbohydrates are the polyhydroxy aldehydes or ketones, and or the substances that yield such compounds on hydrolysis. So carbohydrates are the polyhydroxy aldehydes or ketones, or they can be considered as the substances which upon hydrolysis will give us some polyhydroxy aldehyde or ketone. Most of the carbohydrates have the empirical formula CH2O to the power in the bracket to the power n. So basically, for example, if we will consider glucose, for example, so there the value of n is 6 and the formula for glucose is C6H12O6. There are three major size classes of carbohydrates. So carbohydrates are mainly classified on the basis of the number of units with which it is made up of. So they are classified as monosaccharides, oligosaccharides, and polysaccharides. So the word saccharide is derived from a Greek word saccharon that means sugar. So basically the word saccharide means sugar, and mono, oligo, and poly defines the size of the class. So mono, as the name suggests, is related to the single. So when there is a single sugar moiety or single or simple sugars that are present that consist of a single polyhydroxyaldehyde or ketone unit, then the carbohydrate is a monosaccharide. The most abundant monosaccharide in nature is the 6-carbon sugar D-glucose, which is also known as the dextrose. Monosaccharides of more than 4 carbon atoms tends to have a cyclic structures. These cyclic structures can be a 5-membered ring or maybe a 6-membered ring. Depending on if they are 5-membered rings or they are 6-membered rings, they are called as the pyranose or furanose that we will study later in this lecture. So basically, if I talk about monosaccharides again, so monosaccharides are the simple sugars. They consist of, as the name suggests, single polyhydroxyaldehydes of ketone unit. And the most abundant monosaccharide is the D-glucose, or which is also known as the dextrose. The next class is the oligosaccharide. Oligosaccharide consists of short chains of monosaccharide units or residues that are joined by the characteristic linkages called the glycosidic bonds. So in carbohydrates, 2 units are connected through the glycosidic bonds. So oligosaccharides consist of short chains of monosaccharides or the residues that are joined with each other through the glycosidic bonds. The most abundant are the disaccharides with 2 monosaccharides units. So oligosaccharides may be further classified into disaccharide, trisaccharide, depending on how many number of residues are joined together through the glycosidic bonds. So most abundant of the oligosaccharides are the disaccharides, and as the name suggests, di means 2. So 2 monosaccharide units are joined. Typical example is the sucrose or the cane sugar, which consists of 6 carbon sugars, D-glucose and D-fructose. All the common monosaccharides and disaccharides have names ending with the suffix "ose." So this suffix "ose" suggests like they may be a monosaccharide or a disaccharide. In cells, most oligosaccharides consist of 3 or more units, do not occur as free entities, but they occur in conjugation with some other proteins or lipids. For example, like in glycoconjugates. So they occur in the form of glycoconjugates. And the next class is the polysaccharides. So the polysaccharides, as the name suggests, are the sugar molecules that contain more than 20 or so monosaccharide units. So basically, we see that on the basis of the size, carbohydrates can be monosaccharides in which there is a single unit. Oligosaccharide that may be consisting of less than 20 units. And the most common one is the disaccharide. Out of all the oligosaccharides, the most common one is the disaccharide in which 2 monomeric units are present. And the next class is a polysaccharide in which the polymers contain more than 20 monosaccharide units, or some may have hundreds or thousands of units even. Some polysaccharides such as cellulose are linear chains. Others on the other hand, like for example glycogen, are branched. So basically polysaccharides, as we will study later also, that they may be linear or they may be branched. Example of linear polysaccharide is a cellulose. Example of a branched polysaccharide is a glycogen. So both glycogen and cellulose consist of same units. They are made up of same monosaccharides, that is, they both consist of D-glucose, but they differ in the pattern of the glycosidic linkage, and therefore they have different properties and different biological functions to perform. So this is just the classification of carbohydrates. Carbohydrates are classified into monosaccharides, disaccharides. You can consider it either in the form of oligosaccharides or you can consider it as a separate class because these are the most common oligosaccharides. And then there are oligosaccharides and polysaccharides. So monosaccharides can further be classified whether they consist of a aldehyde unit or a ketone unit. So they can be classified as aldose or ketose. Disaccharides can be classified as reducing or non-reducing. We will see why they are called as reducing and why they are called as non-reducing. The next is oligosaccharides and the next one are the polysaccharides. Polysaccharides can be homo or hetero. If they are made up of the same repeating units, they are called as homopolysaccharides. If they are made up of different monomeric units, different monosaccharides like for example, glucose or some other monosaccharide combines with the glucose to form the polysaccharide, then this is called as the heteropolysaccharide. So examples of aldose monosaccharides include glucose, while the example of ketose monosaccharide include fructose. Similarly, examples of reducing disaccharides are lactose and maltose. Examples of non-reducing disaccharides is sucrose. Oligosaccharides include example maltotriose. Polysaccharides include examples of starch and cellulose, as I mentioned. They are made up of D-glucose. And the next is a heteropolysaccharide. Example includes heparin. So sugars, as I mentioned, they may act as a reducing agent. So this is an example of how they act as a reducing agent. So this is the anomeric carbon of glucose and other sugars. So oxidation of the sugar molecules is the basis of a reaction which is known as a Felling's reaction. So this Felling's reaction helps us in identifying the carbohydrates that are present. So the cuprous ion, that is the Cu plus produced under the alkaline condition, forms a red cuprous oxide precipitate, and in the hemiacetal ring form, the C1 of the glucose, if we can see, the C1 of the glucose cannot be oxidized by the Cu plus 2. So basically, however, the open chain form is in equilibrium with the ring form, and eventually the oxidation reaction goes to the completion. The reaction with the copper plus 2 ion is not as simple as the equation here implies. And in addition to D-gluconate, a number of shorter chain acids are produced by the fragmentation of the glucose. So basically, this is a colored reaction, and this forms the basis for the identification of the sugars. And this shows that the sugars may act as a reducing agent. And in this, besides D-gluconate, other shorter fragments like other shorter chain acids are also produced by the fragmentation of the glucose. And we can see that here the oxidation is taking place at the carbon number 1 of the D-glucose when the glucose is present in the linear form. So this is the ring form of the glucose, like the glucose is present in the ring form, and this is the open chain form or linear form of the glucose, open chain form, which is in equilibrium with the ring form. And here we can see that at carbon number 1, the oxidation occurs. Therefore, glucose is acting as a reducing agent, and upon oxidation, it is leading to the formation of D-gluconate. So now studying monosaccharides, disaccharides, and polysaccharides in details. So monosaccharides, as we know, are the simplest sugars. Therefore, they cannot be further hydrolyzed. So in their generalized formula, X is almost equal to Y. That is, the number of carbon and oxygen atoms are the same. So all monosaccharides occur in either the D form or the L form. The structure of the saccharides is either ring or a straight chain, as I mentioned earlier also. So if it is a 6-membered ring, it is known as a pyranose. And if it is a 5-membered ring, it is known as a furanose. So whenever the structure of a saccharide is present in a 6-membered ring, which is known as a pyrin, the saccharide will be read as a pyranose. And if it is present in a 5-membered ring, it will be called as a furanose. And these pyranose and furanose name were given by Haworth. And basically in the previous diagram also, we can see that in this, it is a 6-membered ring which is present, and it is a pyranose structure of D-glucose. Also, anomeric carbon means the one, for example, here we can see that it is a beta glucose. So it can be a alpha D glucose or beta D glucose depending on the position of the OH on the carbon number 1. So if it is above, then it is the beta form, and if it is below, then it is the alpha form of the D glucose. So this was about the basic. This was the basic of the monosaccharides, and the monosaccharides, as I mentioned, can be classified as either as the aldose or the ketose, and depending whether the monosaccharides consist of how many carbons, they can further be classified as triose for 3 carbons, tetrose that consist of 4 carbons, pentoses that consist of 5 carbons, hexose that consist of 6 carbon, and heptose that consist of 7 carbon. So these trioses, for example, if they consist of aldose, so the example may include glyceraldehyde. Ketose example is dihydroxyacetone. Similarly, for tetroses that consist of 4 number of carbon atoms and an aldehyde group, the example may include erythrose, while for the ketose, the example is erythriolose. Similarly, for pentoses, which consist of 5 number of carbon atoms, the examples for aldose may include ribose and xylose, and while for ketose, examples may include ribulose or xylulose. Similarly, for the ones that consist of 6 carbon atoms, which are known as hexoses, in case of aldose, the example may include glucose and galactose. So we can see here also that in case of glucose, it consists of an aldehyde unit. So if it consists of aldehyde unit, examples may include the glucose and galactose, while the fructose consists of fructose as a ketone as subunit. For the 7 carbon atoms, heptoses, glucoheptose is the one that has the aldehyde functional group, while pseudoheptulose is the one that has the ketone functional group. So classification of monosaccharides is on the basis of the number of carbon atoms and on the basis of the functional group which is present. So on the basis of the number of carbon atoms, they were classified as triose, tetrose, pentose, hexose, and heptose. And on the basis of whether the aldehyde functional group is present or the ketone functional group is present, they were classified as aldoses and ketoses. And these are the respective examples for each of the classes. The next was the disaccharide. As the name suggests, I also mentioned earlier that they consist of 2 monosaccharide units. Examples include maltose, sucrose, lactose, trehalose, etc. All disaccharides are water soluble. They are sweet in taste, so they are also known as sugars. So sucrose is a disaccharide that is made up of a glucose and a fructose. Similarly, lactose, which is a milk sugar also, so it consists of galactose and glucose as their monosaccharide units that forms a glycosidic bond and leads to the formation of a lactose, results in the formation of a lactose. The next example is a maltose that is made up of 2 glucose units. So the disaccharides, the most common disaccharide examples are sucrose, lactose, and maltose. And these are the respective monosaccharides that forms a glycosidic bond, resulting in the following disaccharides. So as I mentioned here also that there is a formation of a glycosidic bond between the 2 monosaccharide units that results in a disaccharide formation. So a disaccharide is formed from 2 monosaccharides. Here the example is shown for the maltose where 2 glucose units, one is a alpha D glucose, we can see here in the alpha D glucose this OH is below the plane and the other one is a beta D glucose in which the OH is in the above position. So this alpha D glucose and beta D glucose basically leads to the formation the glycosidic bond between this alpha D glucose and beta D glucose results in the formation of a maltose. So a disaccharide is formed from 2 monosaccharide units. Here the example consists of 2 D glucose where an OH alcohol of 1 glucose molecule in the right. So basically the OH group which is present on carbon number 4, 1, 2, 3 and 4. So the OH which is present in the carbon number 4, the alcoholic group, condenses with the intramolecular hemiacetal of the other glucose. So this results in the elimination of the water molecule. And so the reaction says there is an elimination of water molecule and the formation of O-glycosidic bond. So the water molecule is released and there is a formation of a O-glycosidic bond, which can be read as a alpha D glucopyrenosyl 1,4, since the carbon number 1 is forming the glycosidic bond with the carbon number 4. So it is read as alpha D glucopyrenosyl. Why pyrenosyl? Because it is a in this, pyreno means 6-membered ring. So this is a 6-membered ring which is shown here. So it is alpha D glucopyrenosyl 1,4 D glucopyranose. So basically in this case, we see that there is a condensation reaction, or there is an elimination of water molecule during the formation of glycosidic bond. And there is the reversal of this reaction will be the hydrolysis, in which the bond will be attacked by the water molecule and the attack by water on the glycosidic bond. So the maltose molecule retains a reducing hemiacetal at the C1, not involved in the glycosidic bond, because mutual rotation interconverts the alpha and beta forms of the hemiacetal. The bonds at this position are sometimes depicted with the wavy lines as shown here to indicate the structure. That may be either alpha form or the beta form. So basically, the most important point from this slide is that during the formation of glycosidic bond, there is an elimination of the water molecule, and this is known as the condensation reaction, resulting in the glycosidic bond formation. And the reversal of this reaction is the hydrolysis reaction, where the water molecule may attack again on the glycosidic bond, resulting in the hydrolysis of the disaccharide. That will further result in the 2 monosaccharide units. So whenever there is a hydrolysis of a maltose, what we will obtain is 2 glucose units. The next is a polysaccharide. So polysaccharides. So in the first lecture, we studied that when we were isolating the biomolecules, when we were studying the biomolecules, there was an acid soluble fraction and there was an acid insoluble fractions. The one with the lower molecular weight were in the acid soluble pool, while the ones that were of higher molecular weight comes in the acid insoluble pool. The acid insoluble pool was forming the pellet, and polysaccharide was one of the components of the acid insoluble pool, and they are another class of the macromolecules. Polysaccharides, as the name suggests, are the long chains of the sugars, and they can be homopolysaccharides or heteropolysaccharides. So as the name suggests, homopolysaccharides that are made up of the similar units. So homopolysaccharides may be unbranched or branched, and they are made up of same units. So same repeating unit is present and in large numbers, more than 20 numbers, and resulting in a homopolysaccharide formation. While on the other hand, there can be a heteropolysaccharide that consists of 2 or several different monosaccharides that may be joined in a straight fashion, that is in an unbranched manner, or they may be arranged in a branched manner also. So basically, heteropolysaccharides may be branched or unbranched, and similarly homopolysaccharides may be unbranched or branched. But the main difference is that in case of homopolysaccharide, the composition is same throughout, and they are composed of only one monosaccharide, repeating units of one monosaccharide. While in case of heteropolysaccharides, 2 or different monosaccharide units combine to form the heteropolysaccharides. So example of a homopolysaccharides include cellulose. Cellulose is a homopolymer that consists of only one type of monosaccharide, that is glucose. Similarly, similar to cellulose, another molecule starch is a variant of this, but present as a storehouse of energy in the plant tissues. So cellulose forms the structural entity. It is present in the cell wall. Starch, on the other hand, is made up of glucose only, but there is a difference in the branching pattern. So, and their functional role varies because of the differences in the glycosidic bonds that are formed between the monosaccharide units. So the function of the starch is that it acts as a storehouse of energy in the plant tissues. Similar to the starch, animals also have another variant called the glycogen, in which are the storehouse of the energy in the animal tissues. So, which are the storehouse of the energy in the animal tissues. So, animals have another. So, what is the difference between the starch and the glycogen? Starch forms the helical secondary structures. In fact, starch can, since it is forming a helical secondary structure, therefore it can hold iodine molecules in the helical portion. While therefore the starch iodine test gives blue color, while the cellulose do not contain the complex helices and therefore it cannot hold iodine, and therefore whenever we will try to stain the cellulose with the iodine, it will not form a complex with it and it will not give a blue colored product. So basically we want to differentiate between the starch and the cellulose. So the starch iodine is blue in color, starch iodine complex is blue in color, while the cellulose do not contain complex helices and hence cannot hold iodine. Similarly, like amylopectin, glycogen is a polymer of alpha 1,4 linked subunits of the glucose. So this is the figure in which we can see that in the glycogen there is an extensive branching that is present, and there is a difference in the glycosidic linkage that is formed. So at one end there is a formation of 1,6 glycosidic linkage, while on the other end there is a formation of 1,4 glycosidic linkage. So basically glycogen is a polymer of alpha 1,4 linked subunits of glucose with alpha 1,6 linked branches. So in the chain there is a 1,4 glycosidic linkage that is present, but on the branching position the alpha 1,6 glycosidic linkage is present. And glycogen is more extensively branched. On average, every 8 to 12 residues are more compact than the starch. Plant cell walls are made up of cellulose. Paper made from the plant pulp and cotton fiber is cellulosic in nature, and they are more complex polysaccharides in nature. They have as they are the building blocks. Like the papers itself, beside carbohydrates, they have amino sugars and chemically modified sugars. For example, glucosamine. The carbohydrates may act as a building blocks also, as I mentioned that they can form conjugates with the amino sugars, with the amino acids or with the lipids, and therefore they can form amino sugars and chemically modified sugars, for example glucosamine and acetyl galactosamine, etc. Exoskeleton of arthropods, for example, have a complex polysaccharide which is called as a chitin, and these complex polysaccharides are mostly homopolymers. So this is the classification of the polysaccharides. The polysaccharides can be classified as homopolysaccharides or heteropolysaccharides. Homopolysaccharides are the storage polysaccharides and the structural polysaccharides. So storage polysaccharides, as I mentioned the examples, in case of plant is the starch, in case of animals is the glycogen, while for the structural polysaccharides, the plant one is the cellulose and the animal, in case of animals, for example, the exoskeleton of the insects is made up of the chitin. The heteropolysaccharides are also known as mucopolysaccharides, and the examples include heparin, chondroitin sulfate, hyaluronic acid, and keratin sulfate. So these are this was about the carbohydrates or the polysaccharides. And so in the carbohydrates, what we study is that they are made up of basically they are made up of carbon, hydrogen, and oxygen. And also we get to know that carbohydrates can be classified on the basis of the size, on the basis of the number of monomer units they consist of. So they can be classified as monosaccharides, disaccharides, and polysaccharides. Monosaccharides are the ones that made up of single subunit, while the disaccharides are the ones that are made up of two monomeric units. The examples were mentioned during the lecture. So different examples, like for example, the sucrose, lactose, and maltose are the examples of disaccharides. Also, we get to know that disaccharides are formed from the two monomeric units that are linked together through the glycosidic bonds, and depending on whether the And also, during the formation of the glycosidic bond, there is an elimination of water molecule. Therefore, this is a kind of condensation reaction. The next one was a polysaccharide. Polysaccharide that consists of uh, two more than 20 monomeric units of the monosaccharides. And these polysaccharides can be homopolysaccharides or heteropolysaccharides. These homo and heteropolysaccharides have various functional roles. They have importance in the storage. They also form the basics of the structural components. Like, for example, in case of plants, cellulose is a homopolysaccharide that is present, and it is an important component of the cell wall. Similarly, in case of animals, chitin is an important component of the exoskeleton of the animals, which is also an example of homopolysaccharides. Heteropolysaccharides example, as I mentioned, include heparin, chondroitin sulfate, keratin sulfates, etc. So this was about the carbohydrates. The next is the lipids. The next the next biomolecule is the lipids. In the first lecture also, we studied that lipids are not strictly macromolecules, but during the process of homogenization, if since the lipids form the vesicles and they come under the acid insoluble pool. So they forms the part of the pellet, but by molecular weight, they are not strictly the macromolecules. So fats, lipids, water lipids, fats and its derivatives are combinedly known as the lipids. And the compounds, they are mainly the compounds of carbon, hydrogen, and oxygen. But the ratio of the hydrogen and oxygen is not 2:1 as we see in case of the most of the carbohydrates. Example, if we'll compare it to from the glucose, so in glucose the ratio of hydrogen and oxygen is 2:1. So lipids also are also the compounds of carbon, hydrogen, oxygen, but the ratio of the hydrogen and oxygen in case of lipids is not 2:1. The amount of oxygen is considerably very less in case of lipids. And one of the important property of lipids is that they are insoluble in water, while they are soluble in organic solvents like acetones, chloroforms, benzene, alcohol, ether, etc. Lipids occurs in the protoplasm as minute globules, and they do not form polymers. Lipids provide more than double energy as compared to carbohydrates. But the instant source of energy are carbohydrates. So in animals, fat is present in the subcutaneous layer, and it works as a food reservoir and shock absorber. Lipid requires less space for storage as compared to carbohydrate because lipid molecules are hydrophobic and they can condense. So therefore, they require lesser space as compared to carbohydrates for the storage. Animals store maximum amount of food in the form of lipids, and lipids, as I mentioned, are micromolecules. Although they come under the acid insoluble pool, also they form the composition of the acid insoluble pool, but they are the micromolecules. So lipids can be classified as simple lipids or homolipids, compound lipids or heterolipids, or derived lipids. So simple lipids or homolipids can further be classified as fats and oils, and basically they include triglycerides, simple triglycerides and mixed triglycerides, or they may be classified as wax. Sorry, it should be wax. So it includes sperm whale wax, bees wax, carnauba wax, etc. The compound lipids or the heterolipids are the ones that they can further be classified as phospholipids if they are present along with the phosphates, and glycolipids if they are present with the sugar moieties. So phospholipids examples include phosphoglycerides, lecithins, cephalins, plasmalogens, phosphoinositides, for example, phosphatidyl inositol, and sphingomyelins. Glycolipids may include cerebrosides, kerosene, prenosin, nervon, oxynervon, etc. Derived lipids are, as the name suggests, derived lipids are the ones. So these compound lipids or the heterolipids can also be known as the conjugated lipids. since they are conjugated with the other molecules. For example, in case of phospholipid, if we will look into the phospholipid structure, so we know that the phospholipid molecule has a hydrophobic, like it has a hydrophobic tail and a hydrophilic uh, head. So basically, the hydrophilic head consists of phosphate molecule, and the phospholipid is a combination of two molecules of fatty acids plus glycerol plus phosphate plus nitrogenous compounds. So compound lipids are basically the conjugated lipids that consist of, beside lipids, they consist of other components. The next one is the derived lipids. So lipids derived from the simple or conjugated lipids are known as the derived lipids. Derived lipids are basically complex in structure, and they are insoluble in water, and they are soluble in organic solvents, just like the other simple and compound lipids. So derived lipids include the examples of steroids, terpenes, carotenoids, and the steroids can further be classified on the basis of the number of carbon atoms that are present in the tetracyclic structure of the ring, like the number of carbon atoms that are present in the overall compound. And examples include cholesterol, coprostanol, cholestanol, or ergosterol. And we know that ergosterols, cholesterols, they form the important like they are important in the they are the composition of the plasma membrane. So lipids, we know, are very important as they are an important constituent of the plasma membrane. The examples of terpenes include monoterpenes, diterpenes, triterpenes, etc. Carotenoids includes lycopenes, carotenes, xanthophylls. They are mainly the colored compounds that provides the color. So this was about the lipids. This was the basic classification of the lipids. Most important property of the lipid is that they are insoluble in water. Also, they are they are the micromolecules coming under the forming the part of acid insoluble food. So this was about the carbohydrates and the lipids. So we get to know that the so basically lipids, in the lipids, we get to know that they are the fats and their derivatives, and they are also, the composition of carbohydrates and lipids is almost similar. Both of them contains carbon, hydrogen, and oxygen, but the ratio of hydrogen and oxygen varies. In case of carbohydrates, hydrogen and oxygen ratio is 2:1 in most of the cases, while in case of lipids, this is not the case. The amount of oxygen is very, very less. It's considerably very less as compared to the hydrogen in case of lipids. And lipids, the most important function of the lipid, that they they play an important role in the storing the food, and also they act as a food reservoir, they act as a shock absorber, they also play important role in the formation of the plasma membrane or the cell membrane in various components. So these are some of the practice questions related to today's session. So the first question is, which one is a reducing sugar? Options include glucose, fructose, erythrose, and all. So we know that monosaccharides are the reducing sugars, and glucose, fructose, and erythrose are all under the category of monosaccharides, and all of them are the reducing sugars. Question number 2 is, a maltose is a disaccharide, reducing sugar, malt sugar, or all? So we know that maltose is a disaccharide which is made up of 2 glucose units. And now what is a malt sugar? So basically malt sugar is another name for maltose also, you can say it is, which is maltose is a disaccharide. Also, it is a reducing sugar, as we see in the example. So disaccharides, we know that disaccharides can be classified as a reducing sugar or a non-reducing sugar. So we know from this figure, we get to know that the disaccharides can be reducing and non-reducing, and lactose and maltose are the examples of the reducing disaccharides. So basically it is a reducing sugar as well. So all of these options are correct. The next question is which is not true about monosaccharides. Which is not true. So are all monosaccharides reducing in nature? So yes, monosaccharides are reducing in nature. Monosaccharides are soluble in water. They do taste sweet, but they are not always ketose. As we see that monosaccharides can be aldehyde or monosaccharides can be ketose. So yes, the option number 4 that says that that the monosaccharides are always ketose is not correct, as monosaccharides can be aldehyde as well. So the correct answer is that monosaccharides are not always ketose. The next question is which one is a levulose? So option number 1 is a glyceraldehyde. Option number 2 dihydroxyacetone phosphate. Option number 3 fructose, and option number 4 glucose. So basically levulose is nothing but it is a fructose. So levulose is another term for the fructose. Next question is which of the following is not strictly a biomacromolecule? So options include proteins, lipids, polysaccharides, and nucleic acids. So in the lecture we have again and again come to the point that lipids are not the biomacromolecules. They are actually the micromolecules. And while proteins, polysaccharides, and nucleic acids are all macromolecules in which the protein is made up of amino acids, polysaccharides are made up of monosaccharide units, as we get to know in today's lecture, and nucleic acid is made up of nucleotides. So lipids are actually not a biomacromolecule. The next question is which of the following is not a homopolysaccharide? Options include cellulose, glycogen, chitin, and hemicellulose. So we know that cellulose and glycogen, chitins are the homopolysaccharide in nature, while hemicellulose is not a homopolysaccharide. So the next question is glycogen is first, it is a polymer of amino acids, it is a polymer of fatty acids, it is a unsaturated fat, or it is a polymer of glucose. So we know that glycogen is a polysaccharide and it is made up of long chains of glucose, and therefore it is a polymer of glucose. So glycogen is a polymer of glucose, and it is not a polymer of amino acids. It is neither a polymer of fatty acids, or it is not a unsaturated fat. Instead, it is a polymer of glucose. So the correct answer is that glycogen is a polymer of glucose. The next question is which one of the following is a polysaccharide? So we know that polysaccharides are the ones that are made up of one or more. So that is made up of more than 20 units of the monosaccharides. So sucrose, options include sucrose, lactose, glycogen, and glucose. We know that sucrose is not a polysaccharide, it is a monosaccharide. Lactose is a disaccharide. Glycogen is a polysaccharide that is made up of, which is a polymer of glucose, while option number 4, glucose, is a monosaccharide unit. So which one of the following is a polysaccharide? So glycogen is the one which is a polysaccharide. In glycogen, since there are two linkages which are present, alpha 1,4 and alpha 1,6, the right hand side of the glycogen is a reducing side, while the other remaining is a non-reducing one. The next is the fatty acids are linked with the glycerol by. So basically, fatty acids can be linked with the glycerol with the help of the ester bonds. So in case of during the formation of the lipids, there is a combination of fatty acids and glycerol, and they are combined with the help of the ester bonds. If I talk about the in case of carbohydrates, the bond which was responsible for the combination of two units is the glycosidic bond. And if I talk about the proteins, different amino acids are combined through the peptide bonds. So all these bonds are very important to remember. So in proteins, the peptide bond is present. In case of, if I talk about the in case of sugars or carbohydrates, glycosidic bond is present. And in case of fatty acids, ester bond is present. The last question is, all lipids are composed of polysaccharides. Are all lipids triglycerides? Are all lipids insoluble in water, or all of the above? So yes, all lipids are not composed of polysaccharides. All lipids are not triglycerides. They may consist of more than more other units, and but all lipids are insoluble in water. So the correct answer is that all lipids are insoluble in water. So this was about today's lecture, in which we studied about carbohydrates, and we get to know about what are lipids and what is the importance of the carbohydrates and lipids. So again, if I go through the lipids and the carbohydrates, so this was the summary. In that, in carbohydrates, monosaccharides are joined by the glycosidic bond. In lipids, fatty acids and glycerols are joined by the ester bonds. Examples of the carbohydrates include starch, cellulose, etc. Examples of the lipids include fats, oils, waxes, etc. Both carbohydrates and lipids have the same elemental composition of carbon, hydrogen, and oxygen, but the ratio of the hydrogen and oxygen may vary in carbohydrates and lipids. Functions of carbohydrates and lipids include that both of them are the energy source. In carbohydrates, also are important structural components. They act as the reserve food. While if I talk about the lipids, they are the energy source. They provide insulation. They are important membrane components, as we see in case of lecithin, in case of cholesterol, ergosterol, or phospholipid itself are the important membrane components that are present. And uh, lipids are also there are hormones also are lipid in nature. So these are the functions of the lipids. So with this, I would like to end today's lecture, and hope you get to know about what are the basics of the carbohydrates and lipids. And in the next section, in the next session, we will learn some basics about the nucleic acids and what are the enzymes and how these enzymes function. With this, I would like to end today's session. Thank you.